Hello friends, we were talking about molecular taxonomy. This is the second part of the video lecture. Uh, in our first part, we talked about 16S rDNA sequence analysis, the ribotyping, ARDRA, and then DNA, RNA hybridization techniques that are the tools for doing molecular taxonomy. Let us have a recap a little bit. Molecular taxonomy, therefore, is a branch of taxonomy that deals with these macromolecules, the sequences of these macromolecules like DNA, RNA, and proteins. And these sequences serve as a character for comparison or for judging homology with existing sequences of different organisms. So they serve as these macromolecules, they serve as molecular chronometers. That is, they are the determiners of the time that when an organism evolved into a newer species. We can have a look, we forgot to take this in our first lecture. We can have a look of a phylogenetic tree. This is a phylogenetic tree which starts from Leuca, last universal common ancestor, about which we actually don't know what was that. This you can consider as the first cell, the progenitor cell, from which all different types of organisms evolved. So on one hand, this Leuca, last universal common ancestor, the progenitor, it must be a bacteria. It evolved into a chlorobacteria group. That is the group that is capable of photosynthesis. And on the other hand, it evolved into, this should be Halo, I think, so Halobacteria on one hand. On the other hand, it evolved into gram-negative bacteria, the cyanobacteria, and these are the time of that. These are even the phylogenetic time, the geological time when this evolutionary event took place, when the sequences, all the organisms, they have a similar sequence, and suddenly at this point of time, these Halobacteria, they got a new kind of sequence, and they developed into a different organism, Halobacteria. The other branch that continued carrying all these groups have up to this extent, they have a similar sequence. But at this point of time, maybe in certain stage of geological history, that three kind of evolution took place, three kind of sequences developed. One led to the formation of gram-negative bacteria, other different types of new bacteria, and other led to the evolution of cyanobacteria. In this way, these branches, the nodal points of the branches, are the evolutionary events when sequence of these DNA changed a little bit, developing into a newer species. So after those techniques, these RDNA-based techniques, there are some techniques like FEM. This is also very much used nowadays for bacterial taxonomy, molecular taxonomy. FEM is about the total fatty acid profile. It is fatty acid methyl ester. So if you take the first letter F, so what is done in this case? The total fatty acid is first isolated. It is first subjected to, since fatty acid is non-volatile, it is made volatile by taking an ester of this methyl. The methyl ester derivatives are fatty acid, they are subject to gas chromatography analysis. So here is the protocol for that. You get the bacterial culture first and then extract the fatty acid, make a methyl ester derivative of them, and subject to gas chromatography. The gas chromatography result is comes like this. There are so many peaks on this graph indicating a particular type of fatty acid of that particular organism. So this kind of data, this kind of graphs are already present in the database, and so this new data will be compared. Compare the pattern of peaks with pattern in the database, and so you can identify the organism. So FEM has become nowadays very important tool for bacterial taxonomy. The one more tool which has come out recently is the MALDI-TOF MS analysis. What is this? MALDI, matrix assisted laser desorption ionization. It is basically mass spectroscopic MS, one type of, and the TOF means time of flight. Actually, it detects the time taken by a molecule to flight within a cathode tube. say this is the cathode tube within which the molecule is traveling. If you load the molecule here with the help of this matrix, they will take a flight from this place to this place, from cathode to anode, and while doing so, the higher molecule will take a longer time and the less molecular weight molecule will take a lesser time. Depending on that, the molecular analysis is possible. The mass can be detected. So this technique has been used. Normally this technique is used for proteomic research, but nowadays it has become a very important tool for molecular taxonomy also, particularly taking the protein of ribosomes. If you take the proteins of ribosomes, the total amount becomes less, and it becomes comparatively easy to compare. And if you subject these proteins to MALDI-TOF, a pattern like FEM will come out. A graphical pattern of this kind will come out, and there is a database of MALDI-TOF MS also. That has been comparatively, that data is a newer one, but you can have a lot of organisms done MALDI-TOF MS analysis there. By comparing your query MALDI-TOF spectrum, you can designate the bacterial species identification by this method also. So these are the molecular tools available for doing molecular taxonomy at present. Thank you very much for your silent listening.